talking about the Last Gift program, lessons learned from altruistic participants enrolled in cure research at the end of life. So thank you for coming. Next slide. So here are our financial disclosures for the content on the web board are listed here. Next slide. And then these are the disclosures for the presenter, myself as the moderator, and the peer reviewers for this particular activity here. Next slide. And then this is the CME. The IAS-USA designates this live activity for a maximum of 1.25 CME credits. So that's good. And the next is that the webinar is also approved for 1.25 hours of ABIM MOC points in nursing, pharmacy, and pharmacotherapy credits. So that's really good. Next slide. Here's the grant. This is generous support for this activity from the following contributors, which is really nice. Next slide. And then this will be on-demand recording and slides from this webinar will be available for the next day, 24 hours. And then for navigating this webinar, a separate window will show the poll questions, which I think you might see. And so we want you to submit questions using the Q&A button. And we apologize ahead of time if we can't get to all of them in the allotted time, but we'll try. And the chat is open, but please submit using the Q&A button. So now it is my distinct privilege to turn this over to Sara, who will share her slides.
Okay, hi everyone, and thank you for inviting me to present my absolute favorite project, The Last Gift. So as part of this presentation, we will learn a little bit about the history of the Last Gift program, including key contribution from the research community and HIV advocates. I would like to talk about the ethical consideration and practical barriers to, setting up a rapid research autopsy program and to do research at the end of life. We will be listing some of the key research findings from the Last Gift program, both our social behavioral part and then our lab-based part. And then I also would like to explain the meaning of the Last Gift program as a participatory research model and how it impacts the life of our participant, next of kin, and the community at large. Okay, here are my pre-test questions. So the first one is, what is your best guess? What percentage of the Last Gift participant will be willing to voluntarily participate in an intervention in HIV cure research at the end of life, despite knowing that there will be no direct benefit for them? So what is your best guess? 20, 40, 60, or 80? And then this is the second one. In studies like the Last Gift program, what is the primary benefit of using a participatory research model? It empowers participants, the window pops over my writing, and families to actively shape the research process, fostering ethical engagement and trust. It simplifies the informed consent process by allowing researchers to make decisions on behalf of participants, especially towards the end of life. It streamlines study protocol, making research more efficient for scientists, and it removes the need for interdisciplinary collaboration by prioritizing clinical outcomes. Okay, so now let's just jump. Okay. I need to go back one slide. Okay, well, anyway, I don't know how to go back, but so probably most of you know that HIV can establish a reservoir very early after acquisition, right? When someone gets HIV, within a few days or weeks, it spreads spreads all around the bodies, not only in blood, but in immune cells in every tissue, right? From the heart to the liver, to the gut, to fat, every tissue you can name it, you can find HIV. And HIV really likes to play hide and seek with the immune system. But even if, people are on antiretroviral therapies and then they go, the vast majority of the time, the virus comes back. And so you understand how, if we want to ever cure HIV, we do need to study how the reservoir behaves, not only in blood, but in tissue. And this has historically been hard. First of all, because in living individual, it's very hard to get tissue safely. Like obviously, we can do small biopsies on the gut or lymph node, but you can't really get a large amount of tissue that we need to do reservoir studies. And most of the autopsy studies that are in existence, they are not designed for HIV cure research. They have poor antemortem characterization. They follow participant only every 3 to 6 months. So we don't really know if participants are on antiretroviral therapy or what happened at the very last weeks to day before that. And also autopsies are rarely performed quick enough. Most autopsy study, they do the autopsy between 12 and 24 hours. And we know that at that point, the quality of the tissue and the DNA and the proteins are already not optimal. So this is how we came with this idea, right? Davey Smith, who is our fantastic moderator, it was like almost 10 years ago, he started having this idea. Why we don't enroll altruistic people with HME who have a life-shortening illness, like, for example, cancer or cardiovascular disease, and then we follow them very closely. We collect clinical data and all of the information, and then we draw blood until the time of death, and then we perform a rapid research autopsy. And there are some rapid research autopsy program in the US. The vast majority of them are for cancer, so very limited. People collect the cancer surrounding lymph nodes. And so basically, when we started with Last Gift, and we wanted to collect all of these tissues within 6 hours from that, it was a huge, heavy lifting to set up such a program. But we knew like the 6 hours, like the accepted window that tissue should be collected and frozen and stored for optimal quality. So why are we interested in end-of-life research, right? Like we're kind of lucky, like a lot of people that live with HIV are extremely altruistic. They have been living with HIV for a very long time. They were part of the epidemics from the very beginning. They still remember how it was where everyone was dying and they lost a lot of their loved one and family members. And so right now, they are older, they are long-term survivor, they start getting sick, and they don't really have a lot of opportunity to participate in research, but they are really eager to give back, to donate back to their community. And this is why this is such an important population, because they want to give back, that they don't have a lot of opportunity. And so there is really a great enthusiasm about studies like the Last Gift. And also, they don't have a lot of expectation of direct clinical benefit. And this is why, creating this cohort as a novel translational research model that can advance HIV cure research. So this is a slide that shows you the clinical information of the participants that have diseased on Last Gift since the inception in 2017. So we enrolled 43 participants. Not everyone got an autopsy. We did lose a few participants, either because we were not notified or because they moved out of San Diego or because they withdraw from the study. So far, I think we did 37 autopsy. As you can see, the vast majority are wild male, which is a limitation of the study, but it's also reflective of the epidemics in San Diego. Most of our participants die of cancer, and they are on study for a variable amount of time. Some people, enroll in Last Gift and then they die very fast. Some of them stay on for several months. Like our median is about 5 months on study. So we have time to collect information and to get to know every participant. So these are the demographic of the people currently living and enrolled in Last Gift. We have 9 participants right now. We are really making, like an effort to reach out to more female, ideally also to non-white participant. We are, collaborating and starting a collaboration with a site in Brazil that is very interested in setting up a similar program so that we can try to diversify our study population. And here is kind of giving you a breakdown of enrollment by sex at birth. You can see, right, we are funded to enroll 5 to 6 participants, but the Last Gift has been so successful that over the last years, we ended up enrolling 10 participants a year, which is double our desired sample size. And we have been doing better, at least with female participants, thanks to targeted outreach. Here is, just to give you an idea about the granularity of the clinical data that we collect. This is our data from one of our Last Gift participants. We do collect all of the historical data, but then really what is key in Last Gift is that we follow people and try to draw blood until most of the time the day before, if not the same day as participant passes, which is unprecedented. And as you can understand, it gives a lot of information because then we can do a direct comparison between blood and tissue at the time of death. This is a picture of our Last Gift rapid autopsy team. So we're really lucky. We have an amazing team of technician and postdoc and student and fellow that are volunteering to be on call 24/7. So every time one of our study participants dies, and it often happens in the middle of the night, our team is on call. Someone calls me usually, and then I have the nice job of waking everyone up, and we all go to the morgue where we all already have all of our supplies ready to go, and we are a very well-oiled machine right now, and we just go through our process. Most of the time, our autopsies are between 3 to 4 hours. So we almost always make our 6 hours mark. We always start with a minute of silence. That's very important to set the tone of the autopsy. And we read an acknowledgement to make sure we honor the gift of our study participant. This is a map where I can show, where all of our tissues get distributed. So after the autopsy is done, the team goes to the lab, and then we have another, 6 hours of work, taking all of the tissue, labeling, storing, preparing them for shipment. And we ship lots of fresh tissue to all of our collaborators. And we know that we are really uniquely the tissue that people get from Last Gift is really high quality. Sometimes we have like 80-90% viability, which is very hard to achieve with an autopsy. I always like to tell some stories about our Last Gift participant. Obviously, every single one of them is very special, but we have 3 participants that agreed to share their name and pictures. Here on the left, we have Tony Bennett, and here he was in hospice with his husband Blake and another family member. He was our Last Gift 1. He was, absolutely the kindest man on earth. He was so patient with us. And he always liked to joke that Last Gift will make him more famous than the other Tony Bennett. And then here we have Mike Danielson. He was also going by Max. He was a publicist working his entire life in Hollywood. And he had the nicest stories that he will tell about all of the famous actors. So I really enjoyed spending time with him. as you can see, he was someone that was extremely fit, and he died with ALS, which is a neurodegenerative disease. And so it was very hard for him, especially at the end where he was barely able to move. And he was a great advocate of last gift, but not only even for ALS research. In fact, we ended up sharing his spinal cord with an ALS team. And then here is Jim. Jim is very special for many of us. He was one of our long-term community advisors, and he was part of the last gift from the very beginning, really as an advisor. And then, as his disease progressed, he decided to enroll himself, and he passed back in at the beginning of the year. It was a very sad moment for all of us, but also very empowering because he really believed in last gift. And just a few weeks before he passed, he nominated the last gift for a big award for community service. And this is where we took this picture. So the last gift has a big social behavioral and ethics component, which is almost single-handed, driven by Dr. Karine Dubé, that some of you might know, and here is her picture. She has been extremely prolific, right? And we really like to think about the last gift as this participatory research model. Like nothing happened as part of last gift that is not discussed with the community or with the study participant. And the last gift will never be as successful if it wasn't for everyone involved. And yeah, like I think that the shape of the last gift has benefit extremely from all of the discussion that we had with the community advisory board that really spent hours and hours reviewing every single document that we were using. So Dr. Dubé, she has been doing surveys and a focus group to try to really, get a sense of how participant and the next of kin and the research team also, and, feel about last gift. And this is one of many papers that she published. Like these are surveys and interview that she and her team has done with last gift participant. And you can see from the title, right? It already sets the tone about what most people think about last gift. Like the recurrent topics are around dignity and altruism and legacy, and really, like as the name say like giving a last gift back to the community. Here are some quotes from last gift participant. We have so many, and we always, we never know which one to pick because they are so powerful. Here you can see, right, a few examples, like my death will not be in vain, like being part of last gift gives me peace of mind, knowing I'm doing something good. Before I die, I have a feeling of joy, feeling of accomplishment, like really, like when we set up the last gift at the very beginning, like the idea was really to collect tissue, right? But then as we kept going, we really realized it's so much more. Like the participant takes so much benefit to be part of the last gift, and that it's like it's almost like an intervention. And in fact, Dr. Dubé also collected quality of life data for most of our last gift participants, and we published a paper last year about it. And really, we were surprised to see that the vast majority of our last gift participants, even if they are terminally sick, even if they are dying, right? The quality of life does not get worse. Like the vast majority of our people, it remains stable or even it improves over time. And obviously we don't have a control group, right? So we only can speculate, but based on the feedback and the quotes that you can see, we really believe that the last gift, the intervention, the community the connection that they do with the team, that puts so much effort to connect with every participant and make them feel special. Like this sense of legacy really help people pass at ease. So the last gift, from the very, very beginning, we realized that the only way for us to be successful is to always have a next of kin or loved one on board, right? Because obviously those are the people that need to call us at the time of death. And so every time we enroll a last gift participant, we require them to assign a next of kin or loved one. It doesn't need to be a family member. It can be a friend or a partner or whoever they wish. And we create a relationship with that person. We also do interviews with them, both before and after passing of the participant. And we maintain very close contact so that at the time of death, they are responsible to call the last gift team and then activate the process. So our outreach team is fantastic. We select every participant of the team, based on their empathy and manners. And Cheryl, who is, our main outreach person, really, she has, every next of kin on her phone. And even, many, many months after the participant passes, she's still in support, like in contact with last gift next of kin to provide support, right? That's a question that we get a lot, even from the community, right? Like when the participant dies, right? Is the last gift just stepping away, or is there any, follow-up with the next of kin? And I do have to say, like, our team does a wonderful job in keeping contact. Obviously, if they want, right? Some participant after, their next loved one dies, they want to move on and they don't want to have anything to do with last gift. And we totally respect it. We had a lot of next of kin that were skeptical at the beginning. They didn't really want their loved one to enroll in last gift. And then they totally changed their mind when they saw how our team was handling it and how how beneficial it was for the participants to be enrolled. So you can see here some of the, words like support and, professionalism and mental and emotional health for everyone of our next of kin. And then Dr. Dubé also worked very close with the rapid autopsy team. You can imagine, right? It's not easy. First of all, you are on call 24/7, and then you have to wake up and go and do an autopsy. And many of us, like many of our technicians, are not used to it. These are like people that have sometimes never seen a dead person. And so we want to be mindful of their mental health and stress levels. So we did also some focus group, and in general, like almost everyone in the team really understands the power of last gift and the gratitude to be able to be part of it. We recently started doing compassion training with the rapid autopsy team, which was very well received, where, it was a 4-week training where people learned about stress management and compassion for the team and for themselves. We are also collecting data before and after that we will publish. And I've seen some of the data, and they are extremely encouraging, right? Like almost everyone in the team loves to be part of the last gift. And, now we did 37 autopsy, and every single time everyone picks up the phone. And as much as I stress, you have to tell me if one time you don't want to come, if you are tired, just, shut down your phone and I'm okay, right? If you don't pick up, I'm not mad or anything. And I'm always surprised that almost every time we were able to pull together a team, because really there is so much sense, even in the team, that this is an important study. Another important point that I wanted to discuss about the last gift. When we started in 2017, or it was even in 2016 when we wrote our first last gift grant, we got a lot of pushback. I'm sure Davey remembers. some of the reviewer were, they were giving us really bad scores and say, this is not ethical and you are taking advantage of people at a very vulnerable moment in their life and they are dying and we should not be asking all of this from them. And here's where the community really had our back. They stood up for us. They wrote email, to reviewers, to other scientists, and especially Jeff and Andy. So I put the picture here, and they were like, how dare you telling me what I can do with my body at the end of the life, right? Like even if we are dying, and they're not dying, they're just, kind of speaking for the community. Like even people that are dying, we're not kids, right? That's very patronizing and really infantilizing to tell us we cannot participate in this research. And this is where we're starting rethinking the concept of vulnerability that has been, very often, used as an excuse to just exclude more and more people from research without even asking them or even without making sure that they were okay with it, right? Like if we trust the informed consent process and assuming people obviously are not impaired, I do really believe and strongly believe that people should have the opportunity to enroll in a study. So we did wrote this opinion piece that you can see here on the top right, and I highly recommend to read it because I learned so much while we were writing it, and about how much the community of people with HIV have contributed early on in the epidemic and now again to really reshape the concept of vulnerability. So when a population is vulnerable, we shouldn't just exclude them for research. Really, we have to see that as an opportunity to sit down with community leader and decide and discuss together how can we make sure that this participant can enroll in the study safely. So just something to think about, because I see this all the time, even when we design clinical trial and then we start listing, who should we exclude? And, we should exclude women or, why? Let's just think about it, right? If we have a robust informed consent process, I want, in an ideal world, participants should always have something to say about in which study they want or don't want to enroll. So I highly recommend reading this opinion piece. Okay, so until now we talked about last gift, like history of last gift, the ethical and social behavioral part and community engagement and some of the, presenting some of our participants and how the team has really done a good job in creating this participatory research model that has become almost a little bit of a gold standard now. Like the community often refers to the last gift as, a group that did a very good job in setting up a relationship with them based on trust. But now I wanted to kind of present some of the tissue data, obviously, right? Because a very important part of the last gift is the tissue distribution and all of the cool data and knowledge that we can gain and all of the cool paper that we have been writing around tissue reservoir. And we are very, very collaborative. We are actually really committed to maximize the gift of the tissue and the participants. So I always encourage everyone, if you have a project in mind, reach out. If we can help, we will. Okay, so first step, quantifying the HIV reservoir, right? The first thing that we always do in all of our tissue is to extract DNA, extract RNA, and measure the reservoir, right? And then we can compare it between all of the tissue. These are two participants on the top and the bottom. You can see this particular participant has a very high reservoir in the pancreas. So, and this is a very interesting case because he had a pancreatic cancer, right? So this participant here, he had high reservoir in the gut and in the lymph nodes, which is a little bit more expected, right? Like that's where the reservoir is usually found. So you can see, right, there are some pattern in reservoir distribution, but there are some outlier, and some people have high reservoir in unexpected places. And right now we're measuring the reservoir in many more sites. And here, again, to give you an idea about the granularity, these are HIV DNA and RNA level only in the brain. And you can see how many brain sites we collect, right, from the basal ganglia and all of the cortex, hippocampus, medulla, pons, and the spinal cord across multiple segments. And then for every tissue, we extract DNA, we extract RNA, and we measure the size and the activity of the reservoir. And so there is still so much to do, right? All of this gray are, we're still working on it. But yes, so the last gift, like our goal, one of the goals is really to try to map and give a good idea of where the reservoir is, and if there are some pattern of some organs that have higher reservoir than others. Then the next step is we sequence the virus in each tissue, and then we can use this sequence to correlate and see how the viruses across various tissues relate to each other using the genetic information, right? And then we can say, we can answer a question like, for example, is there a specific different virus in one compartment, right? It's called compartmentalization. Or is the virus in the brain the same as the virus in the gut? Or is the virus in the gut the same as the virus in the spleen? And here you can see it's an example of a phylogenetic tree where every dot is one virus, and then you have various color for all of the various tissue compartment. And for example, you can see here, there is one virus that is basically a clone of, a lot of clonal expansion of one virus that is all across the tissues, because you can see all of the dots have different colors, right? So, and this participant has been on antiretroviral therapy for a very long time. And so this, for example, makes me think that even if the virus doesn't replicate, the reservoir is not kind of a stable situation, right? There is still movement of cells with integrated provirus that goes from one tissue to the other. So I think that this is, that was kind of a very nice way to show the reservoir is not stable. Cell can migrate and still bring provirus from one tissue to the other. So Dr. Shayan, who is one of our colleagues, and he has a lot of experience in statistics and bioinformatics, and he used this high-level statistical modeling to try to infer or predict migration event from one tissue to the other. And then he comes up with these very nice figures. And this is a participant that is on suppressive antiretroviral therapy. And you can see that it seems to be that there are migration events from one tissue place to the other. And you can see, for example, here we can follow, right, from the lymph node to the liver, and even from the brain to the prostate, right? It really looks like it's a very dynamic picture. And then we can also use this sequencing to try to predict if a virus is susceptible to some of the therapies that we use as part of HIV cure intervention, like, for example, neutralizing antibody. And in this particular study, it's our team, and always with the help of Dr. Shayan, who does all of our analysis, use machine learning prediction model to investigate if the virus across tissue are resistant or susceptible to these neutralizing antibodies. These are all antibodies that have been used or are used in various HIV cure trials. So we were able to get 33 tissue from 7 last gift participants with almost 800 individual viruses. And so basically what he found is that different people have different pattern of susceptibility. Sorry, I never say that word really good. So And most of the time, these neutralization patterns are pretty homogeneous across tissues, but there are some exceptions. And here you can see, like, this is a list for each last gift participant of which tissue has some predicted resistance to antibody. This means only in tissue, not in blood. And you can see that, for example, for 3 participants, it looks like there are some virus in the brain that might be resistant to neutralizing antibody, but not in blood. This means that when we do design a cure trial and we select our antibodies and we maybe look at neutralization in the blood, we might miss something in the tissue. This is why it's so important, right? We can't ignore the tissue. We can't ignore the CNS. Another beautiful study that came out from Last Gift in collaboration with our collaborator from North Carolina, Dr. Yang and Tang. They were very interested to understand if there was a replication competent reservoir of HIV in the brain. So we knew there is some DNA in the brain, but until now there was no evidence, or until recently, that HIV was replication competent. And so there were some people that just didn't believe the brain was an important reservoir. And so Dr. Yang and Tang, they were doing some experiment with a monkey model, but they were never able to work with brain from human because the vast majority of the brain that they were getting didn't have the quality they needed until they found the last gift. So what we have been doing, we have been shipping fresh piece of brain in North Carolina overnight. And so this collaborator, they are able to isolate microglia, and then they co-culture the microglia with T cell from a person without HIV. And then they wait to see if the virus grows. And they were able to grow the virus, and then they were able, using an in vitro tropism assay, to prove that that virus was indeed coming from microglia and not from like T cell contamination, right? So to the best of my knowledge, this is the strongest evidence that there is indeed a replication competent reservoir in the brain of people living with HIV. And not only that, but this model, right, being able to grow the virus from the brain is a powerful model. In fact, Dr. Yang got 2 out of 1 from NIMH now to be able to characterize the virus and to characterize the mechanism, how the microglia, are able to, to have this latent viral reservoir from an epigenetic point of view. So this is probably one of our most successful collaborations so far. For those of you that were at CROI, our fellow, Dr. Mattia Trunfio, had an oral presentation. So what he did, he basically really jumped into the spinal cord, which has been a piece of the central nervous system that has been neglected from many, many studies. I do have to say that extracting the spinal cord is not easy. So not many studies have the resources to do it. So what he did here, first of all, we measured HIV DNA across many brain regions and the spinal cord. So the HIV DNA level are relatively similar across every brain region. And, if we compare brain to blood, blood has higher HIV DNA. And that's expected, right? And then we started looking at HIV RNA transcription. Here, a lot of data are missing, but one, important information that stood out to us is that it seems like the most active reservoir is in the basal ganglia. That's interesting. I don't think that has been reported before. Right now, Dr. Trunfio has been writing a new grant to try to understand the mechanism and what's special about the basal ganglia. And why is the reservoir so active there? Here we can see, another phylogenetic tree. The blue dots is the spinal cord. In this particular participant, we see there is like a very compartmentalized population in the spinal cord. In fact, about a quarter of the participant had a compartmentalized viral population in the spinal cord that was different from the one in the brain. So, I think it's worth studying the spinal cord. It's not the same as the brain. And then doing again, Antoine Chayon's migration model, and comparing it to all other brain regions, we really were surprised to see that there was, a disproportionate amount of viral migration from and to the spinal cord, right? So it really looks like the spinal cord might be an important hoop of HIV dispersion, across the brain and from the brain to other part of the body. So I think it's really interesting, and that's something that we are definitively following up, and just another story of how the last gift provides opportunity to understand how HIV hides and distributes in the body. We did a lot of brain work. This is another collaborator, Dr. Shaletsky, and he also, he's at UCSD, so he can come directly to the morgue, get his own tissue, brain tissue, and then he goes to the lab, he sorts the microglia, and then he's able to do single cell omics analysis. And he was able to detect HIV DNA and HIV RNA in the microglia. And then he compared the cells with and without HIV, and he was able to show that the microglia that are expressing HIV RNA have a more inflammatory phenotypes with increased expression of some chemokine and interferon stimulated genes. So definitely another very nice paper and a story that we are following up. But also really the technique that we are developing to sort the microglia and to work with live microglia, from human participants. That's not an easy thing to do. So like we did a lot of brain work, but that's not the only thing we do. We are collaborating with another team at the Gladstone with Dr. Nadia Rowan, and she's running CyTOF. CyTOF is like a very multidimensional flow cytometry, but instead of only measuring like 10 or 11 different proteins, she can measure like 40. So she's really able to deeply characterize the cells across tissues. And again, this is only possible with viable tissue. You need the cells to be alive to be able to use flow cytometer. And they just published a paper on Cell Reports where they showed that CD4 T cells in tissue, they're not all the same, right? Like I think that's a very big take-home message, that immune cells are different across tissues, and they also express viral sensor and restriction factor that are different and tissue specific, and very often correlate with how big the reservoir is in those tissues. Recently at CROI, Dr. Yachi Ho, this is another collaborator, she's at Yale, and her postdoc, Dr. Eshetu, they presented data from spatial transcriptomics. So spatial transcriptomic is like transcriptomic, like they sequence a lot of all of the genes, but then it's also spatial in the same that you can really see how all of these cells and gene correlate in the space between each other. And here you can see, like we have been sending her lymph nodes. Here you can see all of the different cell types, like B cells here, stroma, T cells in red, macrophages in blue. And then when you look at, the HIV-infected cells, it was a little bit of a surprise. You can see that they are all hiding within the B cell follicle. So she also did some beautiful pictures here from, this is the lymph node of a person that was viremic with HIV, and here is like an HIV negative donor as a comparison. Again, all of the green dots are the B cell follicle, and the red one are the T cells. And I apologize for those of you who are colorblind. I know that's hard to see. And then you can see all of the yellow dot, and this is the HIV again, right? Like this is, you can really see in the space how HIV is hiding in the B cell follicle. Here again, all of the CD8 T cells, the red dots, they are outside. And the interferon response also seem to be outside of the B cell follicle. So it really looks like, the B cell follicle, it's like a fort, right? Where HIV goes and hides, and all of the effector cells are just outside trying to get in, but they can't. And here are macrophagic and dendritic cells. Again, they can't make it into the B cell follicle. And so, and here she put together this graph where you can see like the HIV infected cells and then the distance between the HIV and most of the cells. Again, like HIV infected cells are the closest to the B cells and more far away from T cell effectors, from the interferon response and the macrophage and T cells. So I don't know, these were like, they presented it at CROI, beautiful data, beautiful pictures, and really nice to see, all of the tissue and the spatial coming together. I was really excited about this. Okay, so we are heading towards the conclusion, right? I hope I was able to convince you of how cool the Last Gift program is, and I hope everyone is as excited as I am. Not only about really being able to offer, this high quality tissue to as many people as we can to really try to dig into understanding the reservoir in tissue, but also the framework that we were able to create with our participatory research model, right, close to the community, and working with them at every step to set up a project that is for them, but really for them and by them and with them, so that they feel empowered to participate and challenging this paternalistic impulse to vulnerability that we unfortunately see very often, and also in clinical trial, but in research. One of the big limitations of Last Gift is, the lack of diversity. We do our absolute best, but the HIV epidemic in California is driven by white gay male, and so this is how most of our participants look like. As I mentioned before, we are collaborating right now with a site in Brazil. They are very interested in setting up their own rapid research autopsy model, and hopefully we will be able to diversify our population and learn even more about how the reservoir differentiate. I have 2 more slides, Davey. I wanted to talk a little bit about our future there. Right? So one thing that we really wanted to do, and our community participant and partner are really interested in, is to use the Last Gift as a model to study interventions at the end of life. So basically, like people, they are dying, right? And so they are willing to participate in an interventional trial, maybe neutralizing antibody. And then at the time of death, we collect the tissue and we can really look how do these, drugs distribute and how do they affect the reservoir across the body, right? Because it might be different one tissue to the other. And again, as I said, our community has been talking about this for many, many years. Dr. Dubé, she did survey of our participant, and like, as you can see, like about 60% of our participants will be willing to participate to at least some cure research. Some are more popular than others, but I find this mind-blowing, right? That people at the end of their life, they are so altruistic that they are willing to participate to a study, right? To get antibody infused, or to just because they want to advance the science, because they know they will not get cured. They are still dying, but they can contribute to the science. Okay, so these are just some more picture of the team, our community event. This is a picture of me and Jim just a few days before he died. This is Mike. This is our Last Gift team. We do a lot of social event and really try to be out there with our community. And then obviously the Last Gift is a huge study with a lot of people involved, from the clinical to the outreach to the ethics, the laboratory team, everyone that is on call for rapid autopsy. And me and Davey, we go to as many as we can. Unless we are not here, we are always there with the team. And that's it. Oh, wait, no, I have to go back to the question. Sorry. Oops, I forgot. Okay, well, okay, let's retry the question. The first time, if you remember, how many participants are interested in voluntarily participate in interventional HIV cure trial at the end of life? You should remember, I just said it. Okay. And then the second one, if you remember, was what is a participatory research model? Okay. Huh, interesting. I don't know. I don't think it, I can't remember how was it the pre-test at 60%. So the explanation, okay, here is the participatory research model. On the explanation, yes. Here data suggested about, okay, this one, almost everyone got it right. This is my explanation for number one. It was about 60% for most of the intervention, which is amazing, right? And then for the second one is like explaining how a participatory research model is, where we work together with family and community to try to, shape the research process. And it's very important in end-of-life study, but really, I feel everywhere. We always should try to do it, no matter what type of research we do. I'm done now, Davey.
That was amazing. Always good to hear. So I've seen it so many times, and I love it each time even more. And I actually really like these questions. They were a little hard, but they're good. So thank you everyone for joining. Thanks, Sara, for the great presentation. Hopefully we left people with some thoughts about end of life and how it can be inspiring, especially in the area of HIV and cure research. And I think, is there any other?
A few, so a few people said, wonderful program, thank you so much. So is there any prospective hypothesis before the collection of the tissues? Well, yes, we have a lot of hypotheses, right? We were, like the hypothesis was that, there will be reservoir everywhere. We hypothesizing that we were thinking that the reservoir was the highest in lymph node and gut, which is the case in the majority of the cases, but not always. Like as I showed in that one example, there are participants that have unexpected reservoir that are big in one tissue compared to the other. And we're very interested in the brain. Like we do a lot of brain work. We do believe the brain is, a sanctuary that is hard to reach. And not all the drug penetrate effectively in the brain. So, having a good understanding of how the reservoir looks like there, I think it's very important. And now really another important part, we started with the virology and now we're shifting to try to understand the immunology around it. So with all of our collaborators, and I think one take-home message is the immune response is different in tissues. And, if we try to cure HIV, we might have to tailor our, strategy to every tissue, right? Because it really looks like one cure might not fit all. There is a long question. I need to read it. David, do you have?
I got it. I can summarize it, but basically talking about how do we talk to other procurement organizations and hospitals to increase visibility, donate through life, etc. I think this is really important. But do you have any thoughts around expanding? Because the next question kind of goes along with that in terms of other people who might be interested in the program? And if there are referrals, are you taking referrals along those lines, or any ideas around expansion?
Yeah, this is a very good question. And like we, at the moment, we get, we get a lot of interest around Last Gift, and we regularly get inquiry through the webpage or a phone call from people that are outside San Diego because of the particular requirement around rapid research autopsy. At the moment, we only accept people in Last Gift that are either in San Diego or close to San Diego, because we want to be able to do the 6-hour autopsies. We have been, we are open to help everyone set up a rapid autopsy program anywhere they want. And we have been talking with people, but it's a lot of work. And so usually, they come, they ask, and then, there is not much follow-up. Right now, our biggest hope is the site in Brazil. We have a wonderful outreach team. We are in very close contact with, hospices, with organization, body donation, so that we exchange information. Like a lot of body donation programs don't accept people with HIV. And so when they have requests, they pass on the request to us. There are other autopsy programs that are not rapid, like, for example, NNTC. They have sites, I think in Texas, New York, and LA, if I'm not wrong, and they do accept donations. So if, and there are also, and then there are non-HIV body donation programs throughout the nation. But for us specifically, we only limit, San Diego and LA, Palm Springs, for very specific cases, but we need to be able to, to do the follow-up and get the body. And if you want to refer someone, you can find me or Davey. We are, easy to find, and email, and we will reply to you very fast.
Thanks, Sarah. That was very good. Yes, we will respond and even make connections if we're not available here. Okay, that's the questions. We've got just lots and lots of kudos. Sarah, as always, you do a great job, and I really appreciate it. I think it's really nice to get the word out. So I think we have some more slides.
We have a question. What is your best recruit for reaching HIV community? Okay, so we work very closely with HIV provider. In San Diego, there is the Owen Clinic, which is the biggest clinic and treats over 3,000 people with HIV. And so we are working very close with the Owen Clinic, and that's where the majority of our referrals come from. And we also screen, the hospital records, and if we see one of our participants, in the hospital, we usually check so that we can enroll them. But we do, a lot of outreach. Like I present Last Gift everywhere they invite me, really. Community advisory board, hospices, clinics. And again, like the community here in San Diego is very extremely engaged in Last Gift. So there is also a lot of word of mouth, where a friend of a friend that hears about Last Gift. And so I will say that we even wrote a paper recently about outreach. And I think that our most successful outreach usually is, with the primary care provider, because then they trust their provider. It's a difficult conversation to have, right? End of life conversations are difficult. And even if we are good at it, it's always better if the initial ask comes from the provider. They are more likely to say yes because they trust them and they have a relationship. It's a little bit, that's an interesting fact. We have a arm of people without HIV, and it's counterintuitive, but we have a much harder time enrolling people without HIV than people with HIV, even if there are many, many more people without HIV, and people are dying every day. But I really think there is not the same level of commitment to the cause. And people with HIV are just so, I don't know, for them it's so important to contribute. And the vast majority of the time we approach someone, they enroll. While people without HIV, they are, they don't really care. And so they often say no.
Sometimes. Okay, Thank you. This has been great. I really appreciate it. So some closing moments just for everybody to know. So evaluations and information on how to get the CME and the credits will be emailed by 5 o'clock tomorrow. Your feedback in the evaluation is super important to us so we can continue to improve. A high goal of ours is to continuously improve all of our presentations. And if you're not receiving our emails, please check our FAQ section on our website. Next slide. And next is going antiviral with our famous Dr. Sag. It'll be Innovations in HIV Service Delivery, Building a Path Forward is also on the list. So please subscribe, and then we can get that so you can know when things are happening. And then Doug Richman and Marie White-Winsing is going to talk about basic principles and clinical relevance for drug resistance with the 2025 update. So please don't miss those. Those are two of my really good friends, and that'll always be a good talk. And then we have the Scott Hammer annual update on HIV management in New York on March 24th. So it's coming up soon. Here are the details for that, and it's always a good time. And then we have the annual update on HIV management in Atlanta, Georgia, where Dr. Sag will be doing and that'll be on April 2nd. So that'll be good as well. And then there'll be a virtual course on STIs and HIV prevention and treatment, the hot and the bothered, which is just lovely, with Raifi helping on those lines. So, and our last update is on urgent virus outbreaks with Carlos Del Rio, Peter Chen-Hong, and Yvonne Maldonado. That'll be scary but timely talk. And then future stuff, conference on bacteriophages in DC in October should be excellent. So please, that'll be brand new. And thank you. Thanks everybody for participating and listening, and for all the kudos. Thanks, Sara, the presenters, and of course, IAS-USA for hosting. Okay. Stay well, everyone.